Hi kids, it's me, Mr. Adam, and I am here to give you a presentation on genetics. This is a presentation that I have done in the past with success for kids. It's a lot of fun, I really enjoy it. It's a really good way to teach genotype and phenotype. I'm going to get started by talking about what we're going to do, and then we're going to use buttons to explain genotype and phenotype. This presentation is primarily for, I would say, 5th and 6th graders, but again, anybody should be able to access and learn from this presentation. I love using the buttons here to teach these concepts, so here we go. Genetics comes from the Greek word genetikos, and it is based on the Greek word genesis, or origin. For anybody who knows about the history of religion with respect to the Hebrew Bible and the Christian Bible, Genesis is the first book, and it literally means the origin of things. So genetics is the study of the origins of where we come from. So there's a nice little root word for you. Kids, we study Greek, and here's one you should know. Genetikos, from the word genesis, meaning origin. And hopefully you can read that Greek and figure that out. The founder of the modern science of genetics is a guy named Gregor Mendel, and he worked with pea plants. He was a friar, and he made a lot of discoveries about how traits are passed down from parents to children. There's a lot of definitions that I'm going to give you. Don't be scared off by them, but he used pea plants and the traits associated with peas, smooth or wrinkled, yellow or green colored. He used these traits and he tried to crossbreed them to figure out how the traits were passed on from the parents to the offspring. And in so doing, he figured out about genes and traits, and then of course later scientists built upon what he found to explore the science of genetics, and we ended up learning about DNA and RNA, genes, alleles, the individual characteristics or the versions of the gene. So Gregor Mendel, big shout out to him for his work in Brno in the Czech Republic back in the 19th century. The tool that we're going to be using to talk about genetics is called a Punnett Square. It's named after a scientist, a guy named Reginald G. Punnett. It is a square diagram used to illustrate potential genotypes and phenotypes. So it's a tool that we're going to use to illustrate how a plant might look on the outside, or any organism might look or appear with its traits or characteristics, but also at the level of its genetic code. So the Punnett Square will help us determine phenotype and genotype. Genotype is the genetic makeup of an organism. There's the root gen again, the origins of. So the genotype is what you are at the cellular level with respect to your DNA. So your genotype is actually what your genes make you out to be, with all your different genetic markers and codes, etc. So that is what your genotype is. It's at the level of your genetic code of your DNA and your RNA. So that is what you are fundamentally, genetically. Any organism for that matter. However, how those traits are expressed through the interaction of the genotype with the environment is known as a phenotype. So these are the observable characteristics of a living thing. You cannot see genotype, but you can see and observe phenotype. So I'm going to use the buttons here to illustrate how that works. A few more definitions along the way. A trait is a distinguishing quality or characteristics. So the idea of blue eyes, brown hair, blonde hair. There are many, many different traits. For my examples here, I'm going to be using green leaves versus orange leaves. There's all sorts of traits that we can identify with respect to animals and plants and organisms in the animal and plant kingdom. So a trait is a distinguishing quality or characteristic. Getting back to genotype, genes are the unit of heredity. They govern the traits we receive from our parents. Some genes are dominant, while others are recessive. And we use the term allele to talk about the forms of these genes. Excuse me for a second. So without further ado, let's get started. A Punnett square is something we use to determine how traits are expressed in offspring. So let's say we're going to do what's called a monohybrid cross. We're looking at one trait, and that one trait is going to be represented by a green button and an orange button. The green button I'm going to identify with the letter capital G. Okay, so that's going to be capital G that goes right there, and I'm going to identify the orange button with lowercase g, and we are going to use these to express the dominant trait of receiving green leaves from the parent plant, and the recessive, if you get this allele from the parent plant, you would get orange leaves, even though it's represented by little g. So let's look at all the potential combinations that could come from two parents that both have green leaves. You're going to get, and this is how the Punnett square works, set up so you have a parent that is what is called homozygous, which means they have two of the same, and you have another parent that is homozygous, and I got to put the G there. Let me make a few more here. If we have homozygous parents here, every offspring is going to receive potentially, these are all the potential options for this one characteristic, this one trait. They're going to receive potentially the allele for green from the father parent or the father plant, and another one from the mother. They could also potentially receive the same thing down here. They could receive a positive or a green from the father, but this green from the mother. And the same thing here. They could potentially receive this allele from the father plant and this one from the mother plant. And they could potentially receive this allele from the father and this one from the mother. And that would be the genotype of this plant. This is a homozygous cross here. This is very basic, very simple. And notice, if I stack them on top of each other, you simply have green. This is the representation. This is the phenotype. I'm putting one on top of the other. The rule, though, is that if you're going to put one on top of the other, you have to put a dominant over a recessive. Here we have all dominant, so it doesn't matter. All the children of these two parents are going to have green leaves for this plant. Things get a little more interesting when we talk about what is called a parent that is heterozygous for a trait. If a parent is heterozygous for a trait, that means that they have one allele that is dominant and another one that is recessive. And so in this case, I'm going to create another little g, which would represent the orange leaves here. This is the father parent, or the father plant. Father parent. This is the father plant, this is the mother plant. The father could potentially give the offspring a dominant allele or a recessive. The mother could give potentially a dominant or a recessive. And these are the different four combinations that we're going to get for this trait. The first potential combination is for both parents to give the dominant. And so we have the genotype here of GG. Okay, good game. Then we could potentially get a dominant from the mother, but a recessive from the father. Actually, it's a little bit smaller here. Recessive from the father. And that, for that trait, it would be big G, little g. The rule is you always put the dominant first when you write it out. You could potentially receive from the mother a recessive with a dominant from the father, and that would be big G, little g. And then you could potentially receive a recessive from the father and a recessive from the mother. Now this is what they would look like for this trait on the genetic level with these alleles, right? However, here's the magic part, and this is why I started to use buttons for this. When you show genotype, this is what you have to see. You have to look at the genetic level. These are the two alleles for these traits. Stack, always putting the dominant over the recessive. And what we see is what's going to be expressed when you have the dominant over the recessive. You are going to have what appear to be three out of 4 potential offspring are going to have green leaves. You always have to remember the dominant trait will mask the recessive. The dominant allele will mask the recessive allele, and you will have, of these parents, these two heterozygous parents, you will get 3 out of every 4, this is statistically, 3 out of every 4 of the offspring of these two will have green leaves, and only 1 out of the 4 will have, 1 out of the 4 will have orange leaves. And this is why in nature recessive genes show up a lot less often than dominant genes do. For example, brown eyes are dominant pretty much the world over, and they show up a lot more often than other eye colors in human beings. So this is called a monohybrid cross. Well, let's do a couple of other variations before we move on. What happens if we cross a mother who is heterozygous with a father who is homozygous? Well, let's look at what the potential combinations are going to be. This potential offspring would have this genotype, big G, big G. This potential offspring would be heterozygous, but it would still be, the phenotype would show up as green leaves, but it would be heterozygous. This offspring would be homozygous for this trait, and that would be big G, big G. I need to make another one of those. And this potential offspring would be big G from the father, little g from the mother, and it would look like this. And wouldn't you know it, you have one recessive allele here potentially combining, and genetically these two are heterozygous and these two are homozygous, but with respect to phenotype, all of the offspring of these two have green leaves. So there are many different combinations that we could do. We could do all recessive. This is called a monohybrid cross, and this teaches that genotype is what you are at the genetic level. Phenotype is how those characteristics are expressed and observed. So with the monohybrid cross and these other combinations of the Punnett squares, you can do that with a monohybrid cross here. So there is the monohybrid cross. I would like to turn to something a little bit more complicated because it is so much fun. And I love how we can use the buttons to show how phenotype is what is expressed, whereas genotype is what you are at your genetic level. What I want to do now is I want to look at the potential of 2 traits.